Hi. Hi guys, welcome. So I'm just setting up right now. See if this will work. How are we all doing? Do that. Hopefully we'll do what I need to do. No sound detected. Well, that's a bit strange, isn't it? Oh, here we go. Sorry guys, I was just setting up the other side of things as well. I always start doing lives on YouTube weekly here. Yeah, I'm doing more on YouTube. If you could go through epistasis and autosomal linkage. Addie, absolutely. Miss, I have a question on a multiple choice question. Sure. Let me start with epistasis and autosomal linkage quickly. And then, Rini, if you have the question, if you want to ask away, you let me know what the question is, then I will answer that one. So, epistasis, let's see if I can get this up here. There we go. Good. Right. So that means I can go away from here. I can see it on there. Hi, guys. If you are joining me, welcome, welcome, welcome. Okay. So epistasis and autosomal linkage then. If you're looking at autosomal linkage, the key thing to note is it's giving you the information. It's autosomal linkage. So it's telling you that they are on the same chromosome. So that's the first thing. So I'm just trying to get up so I can actually show you. Here's one I prepared earlier. Because I've got too many screens open, it's going to lag for me slightly. So hopefully I'll be able to sort that. So the topic for autosomal linkage that I'm going to start with is module 6. Rini, what's your question? I am still keeping an eye on that. Aisha, yes, these lives are being recorded. I'm currently on my YouTube channel as and I have the same board shown on my YouTube channel. And you can come and join the live if you want as well. Here we go. So here we go. Right. Autosomal linkage. Took me a while to get the right slides that I wanted. Right, here we go. Paper 2, 2023. Right, give me a second, really. Let me go through this first. So when we talk about autosomal linkage, what we're talking about then is we're talking about chromosomes 1 to 22. So the 23rd pair of chromosomes in humans, we know is not autosomal because they are different lengths. Amna, yes, I will. I've got one other request that I'll do as well, and then I'll come back to that one. So with autosomal linkage, we're talking about anything from chromosome 1 to chromosome 22 where we have homologous chromosomes. So we know, for example, using chromosome 4 here, if we had gene A, we would have a heterozygous individual, let's say, where we have a capital A and a lowercase a because they are different versions. And we've got gene B here, and we would be heterozygous because we have different versions, so different alleles. So when we talk about linked genes, what we're saying is they are inherited as one unit. There is no independent assortment. You know that when you have meiosis, those homologous chromosomes pair, and what will happen is they will then line up at the equator They will line up at the equator in all completely random orders, and they should independently assort if they are not linked. So it doesn't matter whether we have a capital A or a lowercase a or a capital B or lowercase b. Whereas what we find is when you have a linked gene, because they're on the same chromosome, they are one unit. That one chromosome with both of those are going to be moved together as one. So because of that, we know DNA replication, we go from having our chromosomes to having our chromosomes made up of sister chromatids. And then we go from there to potential crossing over occurring. And then potentially, when we have them separated during anaphase 2, the sister chromatids are pulled apart, we would potentially get this chromosome or this chromosome or this chromosome or this chromosome. And these two, because they are different from the parental, they are therefore called recombinants. So when we look at autosomal linkage, that's what we're talking about. Now, epistasis is talking about when one gene is going to have an effect on the expression of another gene. So if I had gene A and that gene codes for a repressor protein, and that repressor protein stops the transcription of gene B, that therefore would be epistasis, where we're talking about one having an effect on the other. Right. So I hope that's helped when you said, Addy, about going over it really quickly. Right, multiple choice question. There is, Chris, so much to memorize. Let me have a quick look at the paper. And see if I can give you the content without giving the question away. I can't remember if they've actually. What paper did you say? Paper 2, 2023, question 9. I need to double check when they've actually launched these papers officially. Question 9, question 9, question 9, question 9. Where are we? Right, okay. Just double check on this one. Okay, so when we're looking at, let me have a look at this question. Okay, so this one's an interesting one. It's the reference to population. Okay, so let me see if I can bring up. Oh. Now, most students struggled with this paper. Hold on, I need you to go away. Oh, I did not need to do that. Let me go here. Right, so if I have very basic one, I'm just going to quickly draw this. So I've got time at the bottom. I've got my viable population. And what you should see is we know that we have got it always following the same population growth curve. We have our lag, we have our log, we have our stationary, and we have our death phase. Hi, Annie. Yes, I will put it on the list and try to get there. If you're looking for specific topic areas, though, please do go and check my videos. They are in playlists linked to your specification. But I'm guessing you've asked about the chloride shift specifically because that's kind of it's there, but I don't go into that much detail, but I will cover it. But to come back to Rinny, your question, what is important when you're looking at this type of question is if we were looking at the population growth curve and we're looking at, let me just remind myself, do they give you numbers? So they give you, for example, lots of different parts here. So there's 1, 2, 3, 4, 5, 6, 7, 8. And we're looking at the fact that in the stationary phase, what that therefore means is the number of births equals the number of deaths. So the fact that it is stationary is because the number of births and the number of deaths are equal to each other. Well, this is log to the 8. So therefore, if we know that that's log to the 8, that therefore means that that number is staying constant at log 8. Whereas if you were looking at, for example, this point here, I'm just putting it at random and coming across, that's only log to the 4. So therefore, that's the reason when it asks you about where you would have the highest death rate, the highest death rate would be the point where it's stationary because the number of births equals the number of deaths. Does that answer your question on that one? So if crossing over occurs, despite autosomal linkage, variation can occur. So if crossing over occurs, so crossing over only occurs where there's autosomal linkage. If there is no autosomal linkage, it doesn't matter, then variation will occur because of independent assortment. So if the crossing over occurs because of autosomal linkage, that's how you will get your variation. Amy, to answer your question, no, I don't think it's going to be harder. I would say, if anything, it's going to be slightly easier. Right, Amna, what did you want me to go through? Oh, 2020. Oh, you're sticking to 2023. Right, give me 1 second. I just need to double check that they have actually published the 2023 papers because I'm not allowed to show them unless they've been published. They have it on PMT already, right? Okay. So if they've got it on PMT, that doesn't necessarily mean, but yeah, it's when they're officially being published, then I can actually then use them. Because technically you're not allowed to, but I believe now we're on to the 2024 ones, which are still under lockdown. The 2023 is okay. Okay. I just don't want to obviously ruffle any feathers. Right, here we go. Chidima, I'm not ignoring you. I'm just trying to focus because the exams are obviously in a couple of weeks. 16 AII, and it was what, paper 1? 2023, paper 1. And 16, 16, 16, 16, 16, 16, A3. Okay. Right. Let me bring this one in. Okay. Done. Right. So in this question, then, you were asked about data from figure 16.1 as well as 16.2. And you were asked about evaluating. So hold on. Go freezing on me now. Give me 1 second. Okay, that's saved. This is why I'm trying to do a million things at once. So because I've got so many different things open, it therefore means my computer sometimes lags. So let me just save my video from my group tuition session. into there, which means I can close that file down. Right, and even better. There we go. Right, so what was 60.1? Okay, so there's two graphs on 16.1. Right. We've got 16.1, we've got this one, and we have got this one. Okay, and then we are asked to look at the following. As cardiac output increased, blood pressure dropped. So there must have been a decrease in vascular resistance. Okay, so they're asking us to look at this data. So the first thing you have to do is really dissect the data. That's the important thing. You need to take the time to dissect that data so you know what it is that you are looking at. Okay, so what I would say to you is whenever you go through a question like this, take the time to look at what they've told us. So, what have we got here? So I've got my stroke volume is on this one. I've got my heart rate on this one. They're both showing you up to 40 weeks. And this one is showing me what's happening at different weeks. And this is my mean arterial pressure. Okay, so as the weeks go on, we can see that there is an increase in stroke volume up until 20 weeks. Then it plateaus off to 24 weeks. There's a plateau there, and then it comes down. What can we see with the length of the pregnancy to the heart rate? Heart rate increases, plateaus here and here, plateaus here and here, but generally increasing until there's a decrease down there. Okay. I shouldn't look to the data. So we've got mean arterial pressure. So we've got a trend that's doing this, but we need to note, it's really important whenever we're doing this, that we're noting the size of our error bars on certain things, because they've given us error bars, and I'm assuming they'll have told us somewhere that those error bars potentially are the standard deviation. Again, it's a case where I have to have a look at the question. Give me a second. They tell us anything about the error bars in the question. No, they didn't actually, but I can make the assumption that they are standard deviation. But okay, they haven't told us anything in the question. So then it's a case of you actually using the information. So as cardiac output increase, so therefore I know that cardiac output is my heart rate times my stroke volume. So I would expect that as the cardiac output is increasing, my blood pressure drops. So you can see that it's dropping down. So from 9 to 12 weeks down to 17 to 20 weeks, the blood pressure is dropping down. And we can see that there is an increase from the 9. So there's an increase here. And there is an increase, but there's that plateau there. So therefore, I want to be giving some information about that. I want to be saying that there is a valid point to this conclusion in the fact that there is an increase in the cardiac output and there is a decrease in the arterial pressure. However, you can quite clearly see the trend goes down and then back up again. Whereas when we're looking at the stroke volume and the heart rate, they're still increasing upwards. So therefore that goes against that conclusion. And then you'd want to be giving information like my error bars are overlapping. I don't have any information on statistical tests. They haven't told me anything about that. So that's where that answer would be coming in. Right. So what's vascular resistance? Are you meant to know it? So no, what they're wanting you to understand is that as the cardiac output increases, the blood pressure dropped. So there must have been a decrease in vascular resistance. What they're actually saying is that first part of the sentence, do you understand that as that one's increasing and that one's increasing, this should be dropping completely, but it hasn't. It's dropped and then risen up. So the resistance, the vascular resistance, is talking about your blood vessels and talking about the fact that you are going to have a larger vascular resistance if you have got a higher pressure. So they're saying that as the cardiac output increased, the blood pressure dropped, so there would be a decrease in vascular resistance. Well, yes, that is true, but it's not true to this. Decreased vascular resistance could explain the drop in pressure. However, what caused the increase in pressure? So it's about can you evaluate the information that they've given to you and then use that to explain what's happening. So Fizza, to answer your question, what does it mean by error bars overlapping? If you have error bars overlapping, then that would give you an indication that there is not a significant difference. So whenever you plot out your data, you plot your data out with the assumption, if I take that away and just put, that's my mean, that's my mean, that's my mean, that's my mean, that's my mean. The only difference is they drew it as a bar chart, right? That's the only difference in the data that they did. I'm just going to take the bars away and just do it as showing my mean point. And then what I'm going to do is I'm going to do my error bars, which is my standard deviation up and down. So if I were to look at only 2 points here, I'm going to look at those 2 points and only those 2 points. So if I look at point A against point B, and again, I'm going to take those error bars away. Right now, if I just look at those mean points, I can see that they are different. So the question would be, are they significantly different or not? So the first thing we do is we look at those error bars, which is the standard deviation, and that is 68% of your data. The larger the error bar, the larger the standard deviation, the more variability there is within your data. And so that tells you that although I've plotted my mean point with that red cross there, actually, how true is that mean point when my data is quite spread out? So when you do your error bars with your standard deviation, you're bringing in outliers as well, because the outliers are kept within all the data. You don't remove an outlier when you work at standard deviation. You show the variability in your data. So basically, whenever you're doing this, what you're trying to show, if I bring in 1 second, my where do I want to go? What do you want? If I bring this in, she says, oh, there it is. So whenever you are looking at this, where do I want to go? Where do I want to go? There we go. I'll give you a little snapshot from my workbook, my study guide. So there you go. If you're looking at any data and you're looking at the standard deviation, then what you want to do is you want to understand that a small error bar indicates a small standard deviation, therefore high repeatability, trying to use those keywords, increased precision in my data. And you can then be confident that the real mean is close to your calculated mean. If your error bars are overlapping, it's going to suggest there's uncertainty. Because it's suggesting there's uncertainty about the difference between those mean values, that's why you want to do a statistical test. So you want to do a statistical test to prove whether or not that is the case. So does that help? You are absolutely welcome. Right, let's have a look. What was the second question you asked me for, Amna? It was 19 something or other, wasn't it? Oh, Paul, I didn't even notice that you'd popped in here. Hold on, 19A. Let's have a look. Right, where are you? 19A, 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 19A. Okay, 19A. So we're talking about a second. There we go. Whether it's true when it comes to your, does that mean sweating? Hold on, let me have a look at the question. What's the question saying? So the question is saying, oh, where am I going? I've lost my PowerPoint for you now. There you go. Right. So we have got, there we go. Maybe up there. The liver, kidneys, lungs, and skin are all involved in excretion. Right. Okay. So what's excretion? So whenever you've got a question like this, the best thing to do is to try and help yourself and say, well, what is excretion? So excretion is the removal of metabolic waste products. So the liver, kidneys, lungs, and skins are all involved in excretion. So in reference to skin, does that mean sweating? Well, yes, because you're going to be sweating and you're sweating out your salts that are included in the water. Carbon dioxide formed by deamination of excess amino acids. Well, obviously that's not true. That's false because it's not carbon dioxide. Urea is less soluble and less toxic than ammonia. Yes, of course that's true. And the breakdown of heme from hemoglobin produces bile pigments. Yes, that is true as well. Right, 19C. 19 CCC. What did you want? C1. Oh, okay. Right. So this one is looking for okay, so in this one, we've got our Bowman's capsule, we've got our proximal convoluted tubule going down to lip of Henle, distal convoluted tubule, and we've got our collecting duct. So inside here, we've got our glomerulus. So they have said that this is W, this actually slightly further back is X, this is Y, and here is Z. And they are asking for where, for part one, why is it Y related to ADH? ADH increases the permeability of the walls. Right, okay, so the reason is not, you've got to remember to cut across. Here's my cortex. Here's my medulla. So therefore, where is it going to actually have an effect? So ADH increases the permeability of the part in the medulla. So ADH affects the collecting ducts in the medulla. Oh my word. Try that again. So does that help you on that one? So why isn't it in the cortex? Because the cortex is the place where you're going to have the ultrafiltration and the selective reabsorption. The whole point in the collecting duct is to remove the fluid which has been put there from the loop of Henle to reabsorb that back into the bloodstream. So the water reabsorption is very minimal, but it's actually it's that whole thing of water moving in both directions. So it's a very small part of it, but it's not to do with ADH. So when we're looking at it with the proximal convoluted tubulate Y, what you're talking about is the movement of salts. And because you've got movement of those mineral ions, that's going to affect the water potential. And that will therefore affect the water moving by osmosis. But that's not actually where ADH works. So ADH, remember, is a hormone that's released by the pituitary gland, which is going to go and bind to receptors. Those receptors are in the collecting duct. So the distal convoluted tubule won't have ADH receptors to increase the permeability of the walls. That's why it's the collecting duct. Yes, but there will be some water reabsorption, but that will be because of osmosis and because of the changing of the water potential. So Addy, I'm not sure if you are still in here. You asked a question about gene expression and hormones. I will come to that in one sec. Was there another question you had on this one? Oh, and the part two of this question. Let me have a look. Oh, okay. So in this one, this next question, it's asking, you put potassium ions, not chloride. Ah, right. Okay. So it's not about, you should know that when we're looking at the ascending loop of Henle, this is where our sodium ions are going to be moved. Because the sodium ions are going to be moved out of the ascending loop of Henle, chloride ions follow suit. So you put potassium. If you put potassium, they will have allowed, hold on one second. Yeah, they will allow an error carried forward, but only once. What about the sodium potassium pump? So the question specifically was asking you about the process in the loop of Henle that causes the solute concentration to increase. The sodium potassium pump isn't going to cause the sodium concentration to increase because remember the sodium potassium pump is to do with sodium moving out and potassium moving in. So if you said about potassium, you're making the potassium or you're thinking about the potassium going in the wrong direction. Because if we're looking at the sodium potassium pump, we know that you will have sodium being pumped out, potassium being pumped in. So therefore, if this is my membrane, this is my cell. So when you talk about how does it actually affect the concentration of ions here, and then if you're talking about the concentration of ions inside the loop, the inside of the loop is because you are pumping the sodium out, water is moving out and of the descending limb, because the water is moving out of the descending limb, what it does is it makes it more and more concentrated as you go down. So if I, hold on, let me bring in this one. Um, if we're looking at oh, went too far. If you're looking here at the loop of Henley, you can see this is the solute concentration, this is the solute concentration, this is the solute concentration. So because we're pumping the sodium out, chloride will move with it. What we're creating is a high salt concentration in the medulla. Therefore, water is going to move out of here. So because the water is moving out, the salt is going to become more concentrated as we move down, all the way down to that loop. And that's what's causing the effect. Has that helped? So the chloride follows, basically. So because of the positive charge of the sodium, the chloride will also kind of follow because of that attraction between the two. But we don't tend to talk about the chloride. Yeah, it's all about the sodium and chloride, nothing to do with the potassium. Question 19 from, oh, okay, there we go. 2018 paper 2. Yes, I can. 2018 paper 2. Hold on, give me a second while I find it. What did you say? 2018 paper 2. Right, and you want question 19, right? Question, oh, hold on, where are you gone? Question 19. Right. Oh, crumbs, I did go too far. Which one of question 19 are you looking for specifically? Is it the dilution one? Yeah. Okay, so I actually really like this paper. Oh, no, did not mean to do that. I never thought I would say that about certain question papers, but the more you do it, the more it becomes easier, but it is important for you to note the information that's been given to you. Okay, so I'm just going to quickly put some stuff on here. You've been given so much information on this question that sometimes it can become a bit overwhelming, is the one thing I would say to you. So here's the question that you've been given that you want me to look at. And what they want you to do with this question is they want you to work back and they want you to figure out so I'm assuming it's this one here that you are really wanting to know how to answer the question of. So this one here, how to work it out. So I would do my little thing that I now got. So I've got the number of cells and the volume. So the first thing I would do is I would say, right, okay, so where are we starting with? They want us to do tube F. So here's tube F. So therefore, the first thing I'm going to do is in my plate, oh, crumbs, done it the wrong way around. Let's try that again. In my plate, I have got 2, 4, 6 cells because I'm counting the number of viable colonies that I can accumulate back to being cells. So in my plate, I have 6. Now they've told me that I put in the previous question, they've told me in each of my tubes, A to F, 0.1 of the mixture was cultured. So 0.1 centimeters cubed is my volume. So therefore, here I've put 0.1 centimeters cubed. Now, they've also told me, if I go back up, in the one where I took the 0.1 centimeters cubed, I had 1 centimeter cubed of E plus 9 centimeters cubed of sterile water. So therefore, in here, I had 10 centimeters cubed, one which was made up of the one before, and 9 which was the added water. So 9 centimeters cubed plus 1 centimeter cubed. So I had 10 centimeter cubed. So therefore, I need to divide 10 by 100 to get to my 0.1 centimeters cubed. So I'm going to work my way back to figure out what's in F. So therefore, I'm going to go backwards. And what I need to do is times this by 100. So therefore, in flask F, I had 600 cells. So now I'm going back up to the one above. So now we're going to go to E. Now in E, I, again, because it told me before, had 10 centimeters cubed. Now only one of them, 1 centimeter cubed, was put into F. So therefore, I divide 10 by 10 to get me down to that 1 centimeter cubed. So to go backwards, I'm going to times by 10. So here I have 6,000 in that one. Same again, I have 1 centimeter cubed this time of my D. And because I've got 1, so basically to go from here to here, I need to times by 10 from here to here. I need to times by 10 from here to here, times by 10 from here to here, times by 10 from here to here, times by 10. But remember to go from tube F to plate it, I also had to take this number and times it by 100. And that's where most students miss the part. They can do this times 10, times 10, times 10, times 10, times 10, but they forget that tube F was actually only 0.1 of the original. So therefore, you basically make your way back. So I'm timesing by 10. So 60,000 EC, I'm going to times by 10. So I've now got an extra 0. B to A. So again, I'm going to times by 10, 6, 0, 0, 0, 0, 0. How many zeros have I got there? Yep. And then I'm going to go back up again each time. So I hope that helps. Guaranteed serial dilution's coming up. How it's going to come up, I don't know. But every year, serial dilution comes up in at least one of the papers. So I would definitely say that that is the case. So the 100, and that's the bit that most students struggle with. The 100 was because I've missed a step. If I actually think about it, I've shown this as a tube E and F, but in here, I had 10 centimeters cubed, but I only took out 0.1 centimeter cubed to plate. So that 6 cells, the 6 colonies that I've counted, was only a very small sample of my 10 centimeters cubed, which was in F. So you need to remember it's about the volume that you're taking out of the one before. Right, 17 AII paper 2. Let me see. And then I'll probably finish on that one. Hold on, which one am I on? Paper 2, did you say, Amna? You are very welcome. Paper 2. Hold on. Can't even remember if I've opened paper 2. Paper 2. 17, 17, 17, 17. Where am I? Right, 17. And which is also a zero dilution question. But I love that. Oh, okay, describe how you'd get from a 20 arbitrary unit stock solution. Okay, so So with AS, once I finish the exams, the when I finish the actual exams, my plan is to open up my group tuitions to AS students who are looking to do sessions for their exams. So yes, I will be offering this once the actual exams are over, but we're so close to the real exams now that we need to make sure we get the level that's needed. So we have 20 arbitrary units. Hold on, I've got to go back to that question. What did it tell me? I've got a stock solution, 250 centimeters cubed, and I want to make up 8 to 2 arbitrary units. Using 8 and 20. Okay, describe how a student could produce an 8 arbitrary unit solution from a 20. So basically, you've got the 20 stock. How much are we using? How much do we want? So we've got 50 centimeters cubed, 250. Place one cylinder for 20 centimeters cubed. So I need 20 centimeters cubed. No, I don't. 50 centimeters cubed of 2AR interplastic regions and stopwatch. Right, okay, so my volume that I need, so this is important, my volume that I need is 50 centimeters cubed. So I need 50 centimeters cubed. So because I need 50 centimeters cubed, I need to make my volume for each of these 50 centimeters cubed. So that's important in itself, that I need to have that 50 centimeters cubed each time. So then what I need to do is work out, if I've got 20 arbitrary units as my starting point, how do I get down to what is required? So if I've got 20 arbitrary units is my stock solution, and I've got my volume, my finishing volume is going to be 20 centimeters cubed. This is my stock. This is my distilled water. So if I'm doing this, I'm therefore putting 20 centimeters cubed in here, and I'm putting 0 centimeters cubed in there. That gives me a 20 arbitrary units. So I want to have an 8 arbitrary units rather than 20, but I still want to have 20 centimeters cubed in my volume. So therefore, if I get my calculator, oh, it's telling me it's time for bed. If I do, I want to have, what was I? That's 8 of my stock solution, and therefore I'm going to put, oh my word, 12 centimeters cubed of distilled water. So therefore, if I'm doing that to get my, I've done 20 centimeters cubed again. Why have I done 20? See, this is where you have to be very careful with, I should have had 50 centimeters cubed. So let's try that again. I was going to say those numbers did not add up. So that is 20 centimeters cubed, 20 arbitrary units. So therefore, 50 centimeters cubed and 0 centimeters cubed of distilled water. I want 8 arbitrary units. So therefore, because I want my 8 arbitrary units, 8 divided by 20 times my 50, which would give me 20 centimeters cubed, and therefore 30 centimeters cubed of the stock solution. So this is where even because I'm getting tired, I want to have a 20 to 30 ratio. So therefore, if I do that, if I get that down, divided by 10, I want 2 of stock to 3 of distilled water. Oh, yeah, that's exactly it. But because I'm getting tired, that's why I'm like my brain switching off. And I would say it's one of those things that I would actually say to you if you are someone who does something like that. So in my head, I was writing the volume, I'd written it above, and yet I wrote a different number. And that's because I know that that's what happens with me with maths. And so because I know that, I always have to go and double check. So I was looking at it going, that's not making sense to me because I was getting 8 and 12. And I was like, that's not the right ratio. So therefore, always go and double check. Guys, thanks for joining me. Have a good evening, and I will try to be back here tomorrow. It depends how tired I am because I've got a lot. I'm teaching for 10 hours tomorrow, so it really depends on how tired I am after my course and my private tuition. But if I can, I will jump on here. If not, I will try.